Now we need to talk about glycogen synthesis. This is going to be a different pathway from degradation. So similar to how we saw different parts of pathways for glycolysis and gluconeogenesis, glycogen synthesis is going to have different enzymes and steps than the degradation pathway. Glucose is activated for glycogen synthesis by the attachment to uridine diphosphate to form the sugar nucleotide UDP-glucose. This formation of UDP-glucose from glucose 1-phosphate and UTP is catalyzed by UDP-glucose pyrophosphorylase. Essentially, in order to actually get glycogen started, we're going to be attaching it to a special extra thing. So instead of it just being a phosphate group or something extra, in order to say this glucose is actually going into storage, it's going to go through a fairly unique set of enzymes and a unique mechanism to do so. To get this mechanism started, the oxygen on glucose 1-phosphate is going to attack the alpha phosphorus, or the phosphorus closest to the ribose ring of UTP. So you're going to get hydrolysis of pyrophosphate, essentially, which will then go to 2 inorganic phosphates, which is the driving force for this reaction. Glycogen is one large molecule. This one large molecule is going to be built on a single protein that is glycogen, which is going to be at the very core of everything. The first glucose is actually linked to a tyrosine on the protein. Then we're going to have glycogen synthase, which is going to catalyze formation of alpha 1,4 glycosidic bonds in glycogen. From there, sugar units can then be added from glycogen synthase. So glycogen synthase is just going to transfer things from that UDP glucose to the C4 hydroxyl at that non-reducing end. So in order to do this, notice that this is all alpha 1,4 linkages. So everything in here is nothing but alpha 1,4 linkages with glycogen synthase. You are getting an oxonium ion intermediate here between having the release from that UDP glucose, which lets it be a good attack for that hydroxyl group on that 4th carbon. Since glycogen has branches, and that glycogen synthase doesn't do any branches, we need something that'll be a branching enzyme. So just like we had a debranching enzyme in the breakdown, we now have a branching enzyme in the creation. The branching enzyme is going to take 6 or 7 residue segments of this growing glycogen chain and then transfer them to the C6 hydroxyl group of a glucose residue. So this does basically just do nothing but make branches. So it's going to make branches in really specific sets, so with those 6 or 7 residues of glucose. As you might imagine, if the degradation is under a lot of control, the creation of glycogen is also going to be highly regulated. And as we saw before between glycolysis and gluconeogenesis, here again we're also going to see reciprocal control. Glycogen phosphorylase is going to be, as we saw before, allosterically activated by AMP and inhibited by ATP, glucose 6-phosphate, and caffeine. If you need a reminder of that, there's a different video to talk about glycogen phosphorylase and its activity in breaking down glycogen. Glycogen synthase, which is essentially doing that opposite, is stimulated by glucose 6-phosphate. So there again, we've got that reciprocal control. Both enzymes are going to be regulated by covalent modification, and both of these are going to be regulated with phosphorylation. Before, when we saw glycogen phosphorylase, there was only one phosphorylation site. Glycogen synthase is fairly distinct and different in that it's got multiple sites to be phosphorylated by protein kinases. Once it's phosphorylated, it actually has a lower activity. So again, this is the reverse of what we saw in glycogen phosphorylase. Glycogen synthase has a lower activity when it's phosphorylated, but is activated allosterically by high concentrations of glucose 6-phosphate. So there's at least 9 serine residues and 4 different protein kinases involved to get this thing phosphorylated. As you would expect, there's something that can dephosphorylate this enzyme as well. That's carried out by phosphoprotein phosphatase 1, or PP1. That will then be able to inactivate glycogen phosphorylase and activate glycogen synthase. So notice that just as the phosphorylation had opposite effects on glycogen synthase and glycogen phosphorylase, when we remove those phosphates, we're again going to see opposite effects. That removing those phosphates will inactivate the phosphorylase and activate the synthase. So one enzyme is going to do the same thing, which is remove phosphates, and that's going to have opposite effects on our two different pathways, one to break down glycogen and one to make and synthesize glycogen. Once it's dephosphorylated, our glycogen synthase enzyme has a really high activity and does not require that glucose 6-phosphate for activity. Since we're still dealing with sugars and carbohydrates, we're still going to have to look into the ability of hormones to help regulate this. So including glucagon, epinephrine, and the glucocorticoids. So insulin is going to be released in response to increased blood sugar. So increased blood sugar is going to release insulin, and that's going to trigger glycogen synthesis. So when there's too much blood sugar in the bloodstream, insulin's going to go. And insulin, remember, will also be stimulating glycolysis at the same time to try to get rid of some of that extra blood sugar. Insulin secreted in the pancreas, and insulin will be stimulating glycogen synthesis as well as inhibiting glycogen breakdown. Glucagon and epinephrine both will be stimulating glycogen breakdown. So again, we see those opposite pairs in our hormones. And just that note in the middle, that insulin is what's responsible for basically trying to maintain your blood glucose. So in between meals, blood glucose is between 70 to 90 milligrams per deciliter. Glucose then rises to 150 after a meal and returns to normal within 2 to 3 hours. So insulin does a lot of different things. Insulin's going to bind again to the outside of the membrane to a particular receptor, usually in the liver and muscle cells, which will then trigger that protein kinase cascade. In that cascade, you're going to get a lot of different things happening. So what you can see in the diagram is that protein kinase cascade will activate your enzymes that will then be able to get our glycogen synthase active. So those lovely kinase cascades. It's also going to affect more than just this though. So while insulin is stimulating glycogen synthesis, it's also stimulating lipid synthesis, protein synthesis, glycolysis, active transport. That's what the protein kinase cascade and that GLUT4 vesicle is. It's showing that active transport. And inhibition of gluconeogenesis and lipid breakdown. So insulin's coordinating a lot of different things all at the same time. So that uptake of glucose is going to provide that substrate for your glycogen synthesis and that glucose 6-phosphate, which will allosterically activate the inactive form of glycogen synthase. So everything about this is aimed at creating glycogen. Taking another look at glycogen metabolism overall. So you can see the arrows going from the bottom going towards glycogen are going to be our anabolic pathways, where we're going to start with glucose and ATP to go to glucose 6-phosphate to glucose 1-phosphate to UDP-glucose to glycogen synthase. When we are degrading glycogen in our catabolic pathway, we've got that glycogen phosphorylase enzyme that will then go to glucose 1-phosphate to glucose 6-phosphate to glycolysis. So at the top, you see those two enzymes. Those two enzymes have their own sets of activation and inhibition. What's done here in the color is red arrows are for your allosteric regulation. So you can see allosterically that you have that reciprocal regulation with glucose 6-phosphate activating glycogen synthesis and that same glucose 6-phosphate inhibiting glycogen phosphorylase. The blue arrows are to show the covalent control via CAMP, dependent protein kinase. Also known as that protein kinase A. That protein kinase A is going to be activated by low blood sugar, which will then phosphorylate our glycogen phosphorylase, which will activate that. In that same way, that low blood sugar signal would still activate that protein kinase, but when we have low blood sugar, we don't want to make more glycogen, and that will then inhibit our glycogen synthase. As we're talking about how the body controls energy, we would be remiss if we didn't also talk about phosphocreatine. Phosphocreatine is a molecule that basically helps to rapidly mobilize a reserve of high energy phosphates. So we don't have a lot of ATP hanging around, but creatine and phosphocreatine are basically the next step. So they're still high energy molecules, and they basically can help replenish ATP stores relatively quickly. You can also have dietary supplements with creatine that can help increase the muscle stores of phosphocreatine and can improve performance during brief intense exercise. The FDA does recommend doctors' approval for dietary supplements with creatine. With increased exercise, you're going to deplete that free ATP, and phosphocreatine is basically going to prolong the ATP availability, but only for a few seconds. So phosphocreatine, again, helps, but it doesn't solve the energy crisis that can happen in muscles when you're exercising a lot. So let's take a closer look at how your muscles actually do work in getting enough energy during strenuous exercise. So there's a lot of different colors on here. So there is this orange line that's very quick and drops off very, very quickly, which is your ATP. So your ATP stores get used up exceptionally quickly, and that pink or red line then is your phosphocreatine, which is helping to replenish your ATP stores. So once your phosphocreatine is used up, which happens in about 30 seconds, then you need something else, which is again where that anaerobic metabolism often comes in. So that lactic acid then gets built up because your muscles need something immediately, something right now. As you keep breathing, something that takes a little bit longer to kick in is that aerobic metabolism. So aerobic metabolism will still help provide energy, but not nearly as quickly as anaerobic metabolism. So it's that longer term exercise that's going to allow for glycogen breakdown and that aerobic metabolism. So looking more specifically at just glycogen stores in exercise, if you keep a really light exercise, your glycogen stores don't change very much. Over 2 hours, they finally start to dip a little bit. But again, light exercise shows very little change in glycogen stores. Moderate exercise, you do start to see your glycogen start getting burned up. But again, it looks like it takes about an hour for that to go down by about half. Heavy exercise then happens faster, as you might imagine. So if we're going to break down glycogen stores with exercise, how long does it take for those things to get built back up? So assuming 2 hours of exercise, with a high carbohydrate diet, your glycogen stores get replenished much faster. With no food, they get replenished at still a very low rate, but faster. And then with a fat and protein diet, they do get replenished eventually, but not, I guess, as you can see, not nearly as quickly as with a high carbohydrate diet. And you notice that with no food, those don't get replenished nearly as completely as they do with the high carbohydrate diet, or even the fat and protein diet. The fat and protein diet just takes a lot more time. Glycogen storage diseases. So there are 3 I want to talk about briefly. 2 of these actually have fairly similar symptoms to them, which are the von Geroch's disease and the Cori disease. They do involve issues with different enzymes. So the first one has that issue with the glucose 6-phosphatase. Because you don't have that breakdown of glycogen, the glycogen is normal in structure, but you have a lot of it because the body can make it, but can't actually break it back down again. So as a result, you get glucose 6-phosphate to accumulate in the liver, and then it can't be converted to glucose, which will stimulate glycolysis, but then causes lactate acidosis. Cori disease involves issues with the debranching enzyme, where only the outermost branches of glycogen can be degraded. So again, you have issues with glycogen being degraded, so you get very similar problems as that von Gerich disease. The McArdle disease then is one where you have an issue with muscle glycogen phosphorylase. So in that case, you have a limited ability to perform strenuous exercise, but gentle exercise is possible. The other thing that we didn't actually talk about in these slides is the fact of why on earth your body actually makes glycogen. So just realize that the whole purpose of making glycogen is the fact that you have one really long polymer instead of a bunch of little monomers. If you have a whole bunch of little monomers hanging around, you're actually going to disrupt the amount of water needed to hydrate all of that, because you're going to have a really high concentration of sugar inside of the cell, which is going to draw in a lot of water to go with it. To a point, that's good, but to a point, that's really bad, because you're going to deal with osmotic pressure problems. So glycogen is also the body's way of solving the problem of being able to store glucose, but not storing glucose as a ton of little monomers. Instead, you store it as one large polymer, and with one large polymer, you don't need nearly as much water to go along with it.